Okay, so the first question in your notes, we basically want to go through and talk about why do we need to make these gamete cells. Remember that gamete cells are like your sperm and egg cells, so why do we need to go through and make these? So take a second, pause, so you have a chance to go through and kind of think about it on your own. But you may have thought of if we, because remember in humans, we have 46 individual or 23 pairs. So if we did not go through and make these gamete cells, then we would have chromosomes that had 46 chromosomes each. So 46 plus 46 would get us our 92. So if this baby had 92 chromosomes, that's double the amount of chromosomes that they need. And if this baby had babies, then they'd have 4 times as much or even more chromosomes than what they would need. So as they keep going down or as organisms kept reproducing, they just keep getting more and more chromosomes. Where we know that gametes are called haploids because they have half of the information as the parent. So because they're haploid, when the 23 chromosomes from the sperm and the 23 chromosomes from the egg come together, they're making an organism with 46 chromosomes, or excuse me, making an organism with 46 chromosomes, which is the same total number of chromosomes as what the parents had. So we need to make these so that way we keep the correct number of chromosomes in the cell. So there's a process called nondisjunction. And nondisjunction is basically what happens if there's an error in meiosis. So remember, meiosis is the process of making gametes. Meiosis is basically taking the now chromosomes and splitting them into those egg and sperm cells. So if that splitting is not done correctly, then we can start to get errors or we can start to get problems. So remember, the steps of meiosis where they're splitting is called PMAT. So prophase, metaphase, anaphase, telophase. If you look at this picture, can I do this one? If you look at this picture over here, you can see that there was a splitting error that was happening. So when the chromosomes line up in PMAT, if they were to split properly, you had one chromosome going one way, one chromosome going the other way, so that you'd have chromosomes that ended up with 2 on each side. However, if non-disjunction happens, then you're going to end up having the chromosomes not split apart. So those cells are not going to have an even number of chromosomes. So this one has too many chromosomes. The last one has too few chromosomes. And because they're not even, we're going to have some type of problems that are happening down the line because organisms either have too many chromosomes and they're making too much of something, or they have too few chromosomes and they're not having the directions of how to make something in our body, which is not good. So here's the normal meiosis. Remember, we're splitting twice, going through PMAT twice. So see this first cell splits correctly again to where all these cells at the bottom have 2 chromosomes in them. Non-disjunction can happen in either meiosis 1 or meiosis 2. So this one's happening in meiosis 1, where we had 3 pairs of homologous chromosomes going in one direction and only one going in the right side. All of the ones on the left, because there were 3 pairs, we now have 3 chromosomes in each of them. So all of these are going to have extra chromosomes, where the ones on the right are going to have too few chromosomes. And we're going to talk about what happens if these get fertilized and whatnot in a second, but I just kind of want to show you what it looks like. And then the other option is meiosis 1 can happen just fine, but there's a problem in meiosis 2. So if it happens in meiosis 2, you can see that the left 2 cells that are highlighted, those ones are completely normal, where the ones on the other side are the ones that are messed up, where one has too many chromosomes, one has too few chromosomes. So let's say these were sperm cells. If the normal sperm cells in this case got to the egg before the non-disjuncted ones, or the ones that had too many or too few chromosomes, the baby would be completely normal and you'd never know anything happened, or genetically normal, I should say. So these two right ones, if those sperms swam faster and got there first, they'd fertilize the egg and it'd be completely normal. But if these ones were to swim faster, fertilize the egg, then you're going to have some type of issue because the baby has too many total chromosomes or too few total chromosomes. If we go through and look, what happens is if fertilization occurs with a gamete, you're going to have an inaccurate or inaccurate amount of chromosomes. The baby's either going to have too many or too few, which is what we were just talking about. If the baby has too few, the baby's basically just not going to develop all the way. It's just going to die, where it may be like a miscarriage, or you may not even know, the mom may not even know she was pregnant at all and just maybe have like a late period. If you have too many chromosomes, that's where you're going to get a genetic abnormality. So that's going to be like Down syndrome is what a lot of us were talking about in prior class periods. So what you guys are going to be looking at today are two things. One, you guys are going to try to figure out what type of genetic abnormality a person has because of the non-disjunction that happened. Before I show you that, though, I want to show you something else. Or just remind, because I should have talked about this before already. Okay, I like this picture. So when you go through and look, we were just talking about, or I was just showing you this picture. So the split 1, split 2. If we go through and look at this picture now, you still have the same type of an idea. So here's split 1. So in this first line, here's split 1. Here's split 2 for the sperm cell. So every early gamete cell for males ends up with 4 sperm. So let's say number 3 had too many chromosomes. If it fertilized the egg first, then we'd have a baby that had some type of abnormality. The opposite side for females is actually, let's go to this one. If you go over here and look at females, females only will have one egg. So every early egg cell in a female only will make one egg, because what they're doing is they're taking all of those chromosomes, or all that energy and all that like cell organelles, and putting it into this one egg, so that way that baby has the most chance for survival. Think about like bigger cell, you have more nutrients, more ways to get the baby energy, more survival. The rest of these things basically just disappear and the body just dissolves them. So when you go back to thinking about non-disjunction for females, if the female had the egg to where this, let me get my little, to where this last one, the one that had 3 chromosomes, if this was the only egg that survived, then 100% the baby's going to have a genetic abnormality. If this one was the only egg that survived, or the ones on the left where they had the normal number of chromosomes, if that was the only one that survived, then the baby will be normal, assuming that a normal sperm comes through. But when non-disjunction happens in females and it's in the egg cell that's ready to be fertilized, it's 100% there's going to be something happening for the baby because of the female egg. Versus if we go back and look at this one, if it happens in one of these male cells, in one of these 4, if 3's a normal one and 3 gets there first, then we're never going to know, or we're not going to know that process happens. But males, just because it happens in a sperm cell doesn't mean that the baby's going to have too many or too few chromosomes. But if the non-disjunction happens in females and it's in the egg that's being fertilized, then it's 100% the baby's going to have that issue, because it's the only egg sitting there. We only have one egg to be fertilized. But yeah. That's one of your questions in your worksheet. I need to go back to my drive. I want to show you today's worksheet for what you guys are going to do after, just so that way you can get an idea of what you need to do. So first questions, 1 through 3, those are stuff that we were talking about in our notes. You can use for number 2, it says to draw a picture. I actually want you to draw. You can go through and draw based on the notes we talked about, or you can draw based on just something you find in Google. These should all be review information, stuff that we've talked about at least 3 times by now. And then this back is the genetic abnormality. So you're going to look at your karyotype over here, this picture. You're going to look at this picture. You're going to analyze this picture. And in this picture, you're going to circle what's different than my normal person. So in here, you're going to have either too many of something or you're going to have something missing. Then for number 8, it says in this chart, you're going to circle which one of these abnormalities they have. So actually in this chart circle, in this picture circle, then you're going to go through and say how many total chromosomes, figure out if it's a male or female. Notice in this chart, some of them tell you if it's male or female. Then you're going to go into Google and just write a paragraph about what it means for that person to have that syndrome or genetic abnormality. So what would their life be like moving forward? There are 5 different versions of this, so you may have different ones depending on the people that are around you, so do not worry or do not freak out if you see someone have a different answer than you.